The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 6th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Wiki Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian, and I've been in the flow cytometry space for over 12 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with an additional location in Europe, we're both here in Philadelphia, and our producer is in Missouri, so we're truly all over the map. We're thrilled to have you listening to the 6th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're amped to have you here, and we are looking forward to a fun episode today. To kick us off for episode number 6, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We'll jump into the latest medical research news.
And Brian, before we do jump into the new news, as it were, I did have a minor correction from podcast 5. So I'd gotten a little bit ahead of myself. Mentioned that MTAGV specifically targets BRAF. And so, essentially what this therapy is TILs that are harvested and by nature recognize tumor neoantigens. At the time of harvest, they're inhibited both by tumor inhibitory cells. However, once they're removed and expanded, they become activated and injected back into the patient. There's more background to that, other things that are happening to ensure the success of the treatment. However, once they are re-injected, then they recognize and destroy the tumor. However, the recognition is based on the neoantigens from having been in the tumor space, essentially. So just wanted to correct that.
Okay. Appreciate that, Adam. Definitely don't want angry cell therapists coming after us for some of these things. But always appreciate that commitment to the details that really matter and making sure that we've got everything accurate as we put that back out into the world. So thank you.
For sure. Mm-hmm.
Right. So in terms of new news today, another round of cell therapy changes. One that we hadn't covered yet was the FDA advisory on the risk of cancer from cell therapies in general. And this was an advisory that the FDA put out in its, relatively dry format that we've got a link to, I think, in the podcast sort of summary here. But this was at the end of November, actually, and it caused quite a stir at the time. We let it settle a little bit just to understand how the field was going to react. But what this is basically advertising is that for all cell therapies generated to date, basically there is some level of risk of additional cancer being caused from these cell therapies themselves.
Right. And so the way they were able to determine this is some of these actual cancers had the chimeric antigen receptor on the surface. And so you were able to determine where it had originated. I think it's important to note again that they did reiterate that the therapies, are for people who are suffering from cancer. And so the risk being fairly minor from the way that they characterize is still present, but it is minor comparatively to the risk of not getting a treatment. And so it is something that they are following up and will continue to follow up for quite some time. And they had mentioned somewhere in 15 years or something like that.
That's exactly right. And, these are very serious indications where, as you say, the risk the risk of not doing anything definitely is much greater than the risk of actually taking these medication. But with pretty much every medication, there is some risk or some certainty of some sort of negative impact. So to have this extremely low, I believe it was less than 0.1% chance of some follow-on effect happening, as opposed to the types of response rates that we've been seeing with cell therapies, it is quite small.
Right. And, certainly on the personal level, there's an impact to it. Right. And basically, by getting this information out, what's being demonstrated is that it's not being taken lightly. It's being investigated. The ideal scenario is not to have any negative impact, and it's just 100% success rate all the time. Certainly, working towards that is extremely difficult to achieve.
Correct. But just like we started out our talk today with a correction about a previous item, if this impact is not listed on the labels, then it's something that needs to be, because it's something that's sort of come up in the follow-up testing. So basically, good that the system is working as expected. The overall industry response is to highlight that it is a very, very low risk. And that the promise of these things definitely still is shining brightly through the additional line item there.
Mm-hmm.
And then that tees up the second item about how this type of risk is controlled in an actual treatment setting. So sort of like last time, I did find a paper as opposed to like a news article, because as flow cytometry nerds, we do read quite a few papers. And this one was on how they're using flow cytometry to monitor for exactly what we just talked about, right? So they're looking at BCMA patients who have received CAR T cells, and they're following up on these CAR T cells to see, how many of these cells, how many of these cells are still there, and B, is anything aberrant going on with these cells? So they talked a lot about sort of developing the panel and how they were using it in the context of these patients.
Right. And so of interest, they utilized a soluble BCMA protein. It was PE conjugated. And so that was the detection reagent. And we're able to get a lower limit of quantitation of 0.19% of CD3 positive T cells. And so the key to this is that it's accurate, it's sensitive, and it's reproducible. So a very useful way to continue to monitor here.
Correct. And what this shows is that basically they've got a very, very sensitive test to be able to identify anything that could be going wrong. So towards the FDA advisory, then this ideally helps to plug this gap. And I think last time we had been talking about the expansion of cell therapies into autoimmune diseases, which in most cases are a little bit less severe than oncology. So in those cases, ideally, this will still be a manageable level of risk.
Right.
So definitely, again, exciting times in cell therapy as we continue to understand more of the risks and how to control them and how to really manipulate them in a number of different disease states here. And moving directly from that into the investment news, I wanted to highlight a couple of news stories. One from Janux, and the other from LB Pharmaceuticals. So Janux is making a T-cell engager, and here their share price shot up 90% on a single biomarker, which I thought was incredibly rapid for just one biomarker as opposed to the primary endpoint of the trial or whatnot.
Hmm. Right. And so this is specifically looking at, let's see, hold on, I want to make sure I've got this correctly. We're looking at prostate cancer, right? And so targeting PSMA that essentially metastatic castration resistant prostate cancer specifically. And so the issue with this PSMA is highly expressed in prostate cancer. And what we have here is a protein based therapeutic with a binding domain specific for PSMA, CD3 binding region. And I was interested when I was doing some reading on this and talking about the CD3 binding region actually being masked. And so looked into that a little bit further, and what the claim is for masking this anti-CD3 region is that it reduces the possibility of a cytokine release syndrome, which is one of the issues you can get when activating the immune system, the T cell, CAR T cells, things of that nature. It can be very dangerous and in fact deadly. And so by masking that, the incidence is lowered. And then finally, they have an albumin binding domain, which my understanding is for that is just a half-life improvement, essentially maintaining this therapeutic within the system longer.
Exactly. And so then, based on that mechanism of action, you can see why this biomarker would have such a significant impact on the share price. And, especially with smaller companies like Janux, with a smaller number of candidates in their pipeline, the sort of progress or lack thereof of a single candidate can make pretty wild shifts in the share price and overall valuation of the company. So
Right.
the joys of biotech, the highs are high and the lows are incredibly low, as we saw at some points in 2023. So
Indeed. And this was all specifically the information I was talking about was Janix 007.
correct. All right. And then the other piece of investment news, I had also pulled out LB Pharmaceuticals. So LB is making a number of different drugs for the neurology space. So schizophrenia is the one that this story was about. And here, the company had been able to raise an additional $75 million Series C. And they were able to do that based again on the strength of their trial results, where they were showing significantly different levels of biomarkers in response to their drugs. And these folks were, I believe, also looking at receptor occupancy or receptor activity levels as a result of their drug.
Right. And if what I read, just understand correctly, this company looks to bring therapeutics from outside the US that have been approved, maybe widely used, and try to bring them to the US for treatments. And so, specifically here for schizophrenia, the LB102 therapy essentially is slightly modified. There's increased membrane permeability so that they could use a lower dosage for it. And right now, it looks like it's widely prescribed in over 50 countries, but just not the US. And it does satisfy an unmet need. Half of schizophrenia patients with current antipsychotics, let's see, less than half the schizophrenia patients treated with current antipsychotics exhibit significant improvements. And so would be helpful if it does end up getting that approval.
Absolutely. And this case as well was critical, basically, with the biomarkers that they selected to show efficacy for this product. So both the LB Pharmaceuticals and the Janix article are just reiterating some of the things that we've talked about on the podcast before. That these flow cytometry investigations can matter pretty significantly as we're going towards valuations and commercialization of some of these drugs.
Yeah, and this LB-102 would be the first benzamide treatment in the U.S. for schizophrenia. So just a little factoid there I just wanted to throw in.
Yeah, nice. All right. And then we can maybe shift from investment news over to KCAS news. And here, I wanted to just, blatantly brag on our team here at the Philadelphia site for KCAS Bio. So our associate director, David Ambrose, did win the Client Champion Scientist Award. So we're extremely proud of this award and of his hard work to achieve that.
Right. So our business team gets together once a year, goes through awarding, things of that nature. And so they do reach out to various key stakeholders within the company to identify the recipients of these awards. And what this means is that David was identified as somebody who is very good in working with clients and, relaying data and making sure that the client is getting what they need, and that projects are moving smoothly. And so I know he takes a lot of pride in those efforts. And so congrats, David. Definitely well earned.
Excellent. All right. Looking forward to a repeat next year. We'll see. All right. So next up is the paper of the month. And here, this also then bleeds into our primary topic of the podcast, which is all about receptor occupancy. So today's paper is called Receptor Occupancy Assessment by Flow Cytometry as a Pharmacodynamic Biomarker in Biopharmaceutical Development. And it's brought to us by Liang et al., which is a team out of MedImmune in California.
And then in cytometry part B, so this is the clinical version of cytometry. There's a part A and part B. Impact factor of 2.65.
So cytometry part A and part B are both critical journals for the field that we always look at very closely. The one is basically pushing the boundaries of what can be done with flow cytometry, and the other is then pushing the boundaries of how that knowledge is then sort of trickled into the field of actually clinical practice. So always good ones for us to use there. And what I like about this paper, it is from 2016, which is older than we'll usually go. That's nearly 10 years old now because we're all the way in 2024, if you can believe it. Despite it being nearly 10 years old, it still is an excellent summary of exactly how important this part of flow cytometry is.
Yeah, and it just demonstrates that RO as an assay has been around for a while in the form of flow cytometry, and like you pointed out, it is very important. Basically, the assay itself has been around. The ways that we go about qualifying and validating receptor occupancy are constantly evolving. We had talked before about H62 and guidelines, and it touches on RO there. What this paper does is really it jumps into. Let me first say there's a lot of information here in this paper. Very good paper, really exhaustive, let's say, and it goes through the biology of our RO to an extent, but then also considerations that you need and how to execute. And so we'll dive into some of that. I think, just backing up within our own experience, we've done RO many times, receptor occupancy assays several times. And what's the first thing, Brian, that you think of when a client mentions RO?
Antibodies and basically this paper and some of the figures that I have seen here, actually.
And I will say, so we don't do this scripted, so I did put Brian on the spot, so I must apologize for that. I will say, for my own purposes, whenever I hear RO, the first thing I hear is in my mind fresh samples, right? And
Yes.
they did address that here, talked about the possible impacts and what happens if you have fixed samples, and is that going to alter the binding, and what the final evaluation of the RO is going to be, and things of that nature.
Yes. There is also that. So, yeah, I mean, when you say RO, definitely fresh samples, definitely pharmacodynamics, definitely antibody binding, and yeah, always a sad thing that we can't use PBMCs in most cases that we've been able to find to really assess this more easily in batches or in a less time-dependent manner here.
And you did mention pharmacodynamic. I would imagine we've got a wide range of listeners, experience with PKPD, maybe not experienced. And so, pharmacokinetic, you can think of as what the body does to the drug, specifically the drug's absorption, distribution, metabolism, and excretion properties. And then pharmacodynamic, how the body is impacted by the drug. And so here, that's what we're looking at, the RO, under the guise of the PD.
Okay. So maybe let's jump from the paper and talk about just the receptors in general. Because the one thing that this I thought was good for the paper was that it simplified the concept of RO enough to fit in a very small figure, right? Where you just see the antibody and is it bound to something and is it glowing or not. But to really get to where we need to be with RO, we need to really understand what's going on with the biology of it. So if we talk about only the biology of receptors for this podcast, and then next podcast we can talk about how to potentially detect this with flow cytometry and validate it, then I think we'll have sort of bite-sized pieces here.
Mm-hmm. Sounds good.
So within receptors in general, there are a few different classes of what the receptor is doing or can do, and how the cell interacts with it. So some receptors have transmembrane sections that can lead to signal cascades and phosphorylation events on the inside of the cells. Some receptors need to dimerize for their functions. Some receptors are internalized as part of the process as well. Other primary classes of receptor types, Adam?
Right. So one of the things that you'll see, type 1 versus type 2 receptors, essentially whether or not the N terminus is buried within the cytoplasm or the C terminus is buried within the cytoplasm. Multi-spanning receptors, thinking there GPCRs specifically. You've got decoy receptors, I guess named decoy receptors. There's always some evidence that they have some sort of function, but essentially a receptor within an extracellular domain, but seemingly not having an intracellular activating domain. And so, there's a multitude of different receptors, but to your point, you have an extracellular domain that is usually bound by some sort of ligand. You have a transmembrane domain as it spans from the extracellular space into the intracellular space. And then you have the intracellular domain within the cytoplasm of the cell, carrying the signaling portion generally of that receptor.
So these are complex molecules then that have complex functions and can then be potentially detected later on as well. So when we talk about receptor occupancy, then we're talking about, is the portion of this receptor, which can be this very large, very complicated molecule, where its ligand usually binds, available to bind new ligands.
Right. And it is important to note that a receptor isn't static. You had pointed out a couple of things where a receptor may dimerize, phosphorylation may happen, and then another intracellular member of the pathway may then translocate to the nucleus for signaling, things of that nature. The receptor itself may change. There could be internalization of the receptor, the recycling of the receptor. And so there is a lot of biology that goes into one of these targets for a drug discovery. And as the paper pointed out, it's important to understand the biology of the receptor. Really to be able to then get to the next step of designing an RO assay that's appropriate for that target.
Right. So maybe let's go into how the drug interacts with the receptor then. And then we can come back to how the receptor can potentially change during transport. So in most cases, the drug is binding to the location on the receptor where something else would bind to. So the ligand for the receptor would ordinarily bind to.
Mm-hmm.
And that can be a competitive binding, but it's usually a relatively loose binding, if you will. It's not like a covalent bond or anything like that that will be forever there. So there's some level of like an on rate and some level of off rate.
Right. And with that, to your point about what the therapy might do, when it binds, it could have certain functions. It could activate the pathway, so it can be agonistic, or it can block or be antagonistic of the receptor pathway itself. And that would essentially get to the function of it. To your point, then it binds the receptor, whether or not that's actually at the ligand binding portion or it's at some other region of it, really depends in part on the function. And then let me think. I had another point, but it eluded me. So let me hand it back to you while I'm recapturing that.
No worries. Yeah, so absolutely. Then some other actions could be taken as a result of that drug binding. But if we think about the sample as part of, let's say, a clinical trial, the
Mm-hmm.
patient is dosed with the drug is interacting with the receptor, the blood is drawn from the patient um, at the clinical site and then shipped to us. So everything that we just spoke about in terms of the receptor activity, the changes to the receptor as part of a normal biology, could be going on during the course of that shipment from the site to the lab. And then, in addition, anything that the drug is making the cell or receptor do as a result of its binding is also going on. So, this is a very dynamic tube that's coming to us at the end of the day. And that's usually what takes about 24 hours from most places in the US to get to a laboratory before it's actually tested.
Right. And so that's where the considerations for the sample matrix come into play. If you're just talking about whole blood, the different anticoagulants could have an impact on the binding there. Then if you're looking at fixation, it sounds like, in a way, fixation should be the perfect thing, right? It just essentially stops everything as a snapshot of when the blood is collected. Unfortunately, it doesn't work that way. A couple of changes happen when fixation occurs, and you could technically knock off some of the therapeutic from the receptor. You could change the conformation of proteins that you're targeting, and so the output looks different. And that's why, obviously, when I mention all about that, we do validations ahead of time to make sure that we understand what the impact of the sample matrix is on the cell type that we're interrogating.
Right. But definitely trying to catch a photograph of a moving target as you are trying to test these cells. So, as the paper said, definitely something that needs to be done quickly and, with as little interference between the drug to receptor interaction as possible. to avoid disruptions there.
Right.
Um, fascinating biology, and then the changes that happen afterwards are also fascinating. So in most of the trials we work on, not only will we have an RO assay, but we'll also usually have a general immunophenotyping assay to capture a picture of the immune system as a whole, and then usually some sort of potency type assay to understand the function of the activated or inactivated cells, as it were, in terms of how it goes. So always interesting to see how that translates from the drug actually taking action on a specific molecule to the cell then carrying out some downstream action and then getting the overall picture of how that's relating to the rest of the immune system there.
Right, and with those additional tests, the decision whether or not you're going to do it in the same tube with the RO, or alternatively, you want to make sure that your RO is nice and simple, and then have the IP in a different tube, so that you're not impacting anything downstream there. And so again, just additional considerations, and I continue to go back to the sample type when you're talking about the receptor biology. And these samples are collected, as you imagine, the samples are at body temperature when they're first collected. You can ship the samples either at room temperature or 4-degree, as it were, to try to make sure that things are maintained as close as possible. During that process, though, these samples aren't frozen, right? So they aren't completely inactive. And so getting back to the actual kinetics of the receptor, is there some sort of internalization or recycling going on where you may get one value RO at the time of collection, but the day later, 2 days later, you might see a different value because of those kinetics. And so, again, those are tests that are evaluated as part of the validation and qualification test scripts specifically.
One thing that we could maybe talk about as a good crossover episode with Dom and company would be how receptor occupancy assays for LBA methods differ from flow methods as well. So it could be a good next episode at some point as well.
Yeah, we've been talking about getting on with the two of them for a couple of months now, probably even longer than that. I mean, we had it scheduled for 2 months ago? And certainly looking forward to picking their brains there and sharing experiences.
Okay, excellent. All right. And then Jeremy, just as an aside, any questions or feedback from people on LinkedIn posts that you've seen?
Not just yet, but we are inviting all of them. Any questions you have, any feedback you want to give, or even a topic that you would like to discuss, let us know.
Okay. All right, excellent. So with that, I guess we'll move on into the end of episode section here and finish out with some weekend plans and I don't know, probably an update on most recent bourbons we've tried.
So before we do that, curious, I know you've got a plan here for upcoming RO discussions, right? So we talked about the biology today, and I didn't want our listeners to think that was all RO, right? You did allude to it a little bit, I believe, at the beginning, that there's going to be more to this. And so what were you thinking about in upcoming RO discussions?
Yeah, so for next episode, I really want to talk about how we validate flow cytometry assays to be able to take into account this complex biology with the receptor occupancy assays.
Mm-hmm.
So I can see that being sort of its own episode. And then I also wanted to spend potentially an episode talking about the calculations and statistics behind receptor occupancy as well.
Mm-hmm. Right. And then within that validation, we would talk about the lead up and looking at the different types of ROs that you can utilize to be able to get the data that you're looking for.
Exactly.
Gotcha. Okay, awesome. And so then jumping back, because you did say something that always interests me, different bourbons. And I will say I picked up Buffalo Trace recently, and after having heard many people talk about it being really good, I was not let down. And I'll be honest, I think that it, at least for me, was on par with one of my other favorites in Angel's Envy. Have you tried that one yet?
I have. I am also a huge fan of Buffalo Trace, so cannot go wrong there.
I'm more of a novice though, so is that just like the standard one, or like what? I feel like there's a whole bunch of different varieties, right?
They do. So they've got a double oaked variety. They have, of course, their standard, and then I think they've got a small batch variety as well, if I remember correctly.
Ah, all right. So more education that I have to get.
Always time with bourbon, indeed.
Yes. All right. And then how about you? Any new ones that you've tried?
I did try, of course, an Elijah Craig recently, and then also a local Pennsylvania one called 4 Grain Bourbon. So that was aged in Jamaican rum barrels right here in the Philly area. So had to give it a try as the local hometown brew, and I was not disappointed. So it was quite good.
Which Elijah Craig was that? Is that the rye or the standard one?
Yeah, that's the straight rye.
Straight rye. That's a good one. I have that one at home too. So awesome.
All right, and with that, I guess we'll close it out here. Thank you, everyone, for listening. Please do reach out if you have any questions or topics that you want us to cover. We're happy to add to the rotation. And ideally, also we'll have a few new guests coming up sometime soon that we'll be able to announce.
Thank you.
Thank you.